So, good morning, ladies and gentlemen. Sorry, I can't speak Bahasa Indonesia, but that's why I present my presentation in English. First of all, thanks for the conference organizers, the Dean of Faculty of Animal and Agriculture and Animal Sciences, the Vice Rector, and thanks for Professor Noor Harini, because of the very nice presentation. It was very refreshing of my knowledge on food technology. And it was a good combination of the probiotics, prebiotics, and microbiome, because these days microbiome is super important. It affects on almost all of the behavior that we have. In English, we have this say that we have, for example, my gut feeling is not good today. Means something wrong in my body, you know. And now you had a good combination that what's happened in the gut, it can affect on everything. You know, some days you waking up and feel that I'm not well today, you know, and nothing happened. It is because of whatever happening in here, the microbes, the interactions with us. Anyway, the title that I choose to present today was for Newcastle disease virus. When Siri invited me or organizing for this conference, and I said agriculture and the sustainability, and I just realized that yes, Newcastle disease is a big threat for the food security in Indonesia. That's why it is well linked with the food sustainability in Indonesia. That's why I mainly focus on it. And I want to introduce a new virus that spread first from Indonesia and then spread in all around the world. And it heavily affects the poultry industry all around the world. So how can I go to the next slide? Oops. So, Newcastle disease virus or Newcastle disease is a disease that for the first time reported in Indonesia, first time in the world, back to 1926, to see where was that. Anyway, I'll go deeper to see where was that in here in Java. And then we go deeper, again, West Java, go to Bogor, and then we get closer to Bogor to see, and the Salak Mountain in there. And I took this photo from the window of one of the hotels that I have, because I love mountains, and it's the Salak. We get closer, we go to the Balitvet, Jalan Balitvet street, and we go to the Balitvet. That's the National Institute for Veterinary Research. So, and by chance, I took this photo with an uncut, and that you can pay, say, 20 cents in Australian money, jump into the car, go wherever that you want. Anyway, we go closer to the Balitvet to see where was the first disease, Newcastle disease in the world, reported in this lab that's established in 1908 by the Dutch scientist, and it was responsible for the surveillance of the animal diseases. Anyway, I took a photo of, from that lab, because you know historically this lab has a great value for the world. So anyway, but when the disease reported for the first time in Java, why the name of that is Newcastle. In May 1926, it was a disease spread roughly between 60 to 80% of the birds, like the chicken, backyard chicken. At that time, it was not the industrial, say, poultry industry in Indonesia, but between 60 to 80% of the backyard chicken, mostly the native chicken, died because of the virus. And because it was heavily impacted in the Indonesian food security, the Dutch scientists packed some samples, sent it to UK for further investigation. And it was a biosecurity failure. because they use the Royal Mail for transportation. And one of the workers there took the box, and he lived in Newcastle. Newcastle is a city in close to at the north, almost northern part of UK. And we feel that the disease is spread by the sample that transported from Indonesia to UK. But because that person lived in Newcastle, the first disease spread in Newcastle, the name of the city. That's why they call the disease as Newcastle disease. But in reality, the disease is say Java disease or the Bogor disease or whatever. To appreciate that, when they isolated the virus, they call it Newcastle disease Java strain. So anyway, it is nothing to do with the Newcastle in Australia. The disease is spread. I don't know why it doesn't work properly. Anyway, the disease is spread all around the world within, say, like 8 to 10 years period. You can see that disease is spread from Indonesia, then to eastern part of Asia, China, India, and then it spread from Europe to the Middle East, and then like entire old world were infected between 1926 to 1933. Almost all old world were infected with the virus. In 1940, for the first time in California, the disease spread. Then in Mexico in 1950, and then both of them of the outbreaks was responsible for transportation of the birds from Indonesia and Singapore to those countries. And then suddenly, by 1966, entire world being infected with the virus. The viruses that reported by that time belong to the genotype 1, 2, 3, and 4. Genotype 1 and 2 was the first isolate that's responsible for the infection, but then they became less infected. Same as the COVID, the COVID compared with the Wuhan strain or the Delta strain compared to the Omicron is nothing. Something happened for the genotype 1 and 2. They are less infectious. And then to see what's happened, genotype 5 and genotype 7 for the first time reported in Pakistan, then spread to Middle East, then almost the Asia, entire world, and then America. Means by 1974, I believe, the entire world being infected with 2 other viruses. We know well that how they make disease, how we can prevent them. In the spreading of the disease was again mainly responsible for mainly happened by transportation of the wild birds from the South America to the North America, or from the Singapore to Europe. Genotype 7 for the first time happened in 1999, and then during a 10 years period just spread all around the world. For the first time, you can see a very small dot, red dot in Java Island there. For the first time, the disease by the new virus reported in Java, and then spread all around the world. Spread in Southeast Asia during a 5 years period, Japan, China, India, Middle East, Russia, Iran, Saudi Arabia, Europe, and then everywhere. America, South America, and then North America, California. So, and California. So to see how this disease is spread, in 2007, we ran a research project on avian influenza in Indonesia, in collaboration with the Balitvet, Gajah Mada University, and funded by the ACR. And we tried to isolate viruses from different spots in Java Island. Unfortunately, it was unfortunately at that time, but that opened a new window for us. all the samples that we isolated were negative. Few samples were positive. But we could see still the chicken in the chicken farms dying because of a disease. And then we try to isolate the virus. As you can see, the mortality rate, the numbers of the birds that dying because of the disease reached to 80%. Means 80% of the birds died because of a new virus. The virus happens in the farms with the good biosecurity and good vaccination. And it was a big question that why we have good biosecurity and good vaccination, but still birds dying. You can see like 80% of the birds died within 24 hours. Like in the afternoon, the farm owner checked everything is good. Tomorrow morning came with almost all dead in the farm. So we started working on it under the previous research project that we had, and we found the serology. Serology tells us the level of the protection. For the level of the protection, we always put a cutoff point. When we say when the antibodies are higher, probably it doesn't work on the as the digital. When the titer is higher than the cutoff point, the birds are protected. But when the titer is less than the cutoff point, the birds are sensitive. We checked them and we found that, the antibody is too high. But the thing is, we use the antigen that is similar to the vaccine, not using the antigen similar to the virus that causes disease. Secondly, we run another experiment. This time we use the virus as antigen that completely similar to the circulating one. And we found that very, very, very surprising results. The birds were not immunized against that particular virus. We call that happening as heterologous, homologous challenges. Means when you want to know how much your body is protected, you need to use exactly the same method for the protection. So we said we used the wrong vaccine, we used the wrong antigen for measurement, and we used the wrong virus strain for the challenge strain. So a paper is published, I think in 2007. Yes, in 2007, it clearly says that when you want to measure the real antibody to the virus, you need to use the homologous antigen. You need to use the virus that's circulating there. Anyway, that's the sequence analysis. I'll prefer to jump over that. That's the usage of the vaccines in Indonesia. As you can see, in the big proportion of the vaccines are Lasota. Lasota is genotype 2, and only 0.04% of the vaccines in the Indonesian market at the current moment is genotype 7. Means we don't have, honestly, we don't have any effectiveness against the virus in Indonesia. Anyway, when we discuss it with the farmers or with the scientists, they said, no, the existing vaccines are good. They can protect it. Of course, they are good and they can protect, but they are not fully protected. They provide, we call it partial immunity. You will see in here that they are the sequence analysis. For the sequence analysis, exactly same as, the thing that's happening for the COVID. They said the virus and the vaccine needs to be matched. They, at the down part of the phylogenetic tree in here, we have the vaccines. And you will see that the biggest distance between the vaccine strains and the G7 cause heterologous, means cause big differences and cause no effective of the vaccines for protection of the Newcastle disease. Anyway, that's a good paper that published saying that if you want to have the proper measurement for the antibodies, you need to use the homologous antigens. Anyway, to see how does it work, that's the virus. When the virus wanted to bind to the cells, they bind through two different proteins. One call it attachment or the G protein, another one is F protein. F protein is like the ice cream cone, the shape of that, and it needs a click to work. Yeah, the virus, that's the animated electron microscopy. It shows that how the virus bind and fuse into the infected cell, to the target cells. Actually, I love that. I show it always in my lectures. So anyway, fusion protein has two parts. And these two parts need to be separated for initiation of the infections. So if those two parts separate easily, the virus will kill the birds. If those parts do not separate easily, the birds will be alive. So that's a fact in vaccinology, or making the vaccines for Newcastle disease virus. We will use it later on. To understand how the disease is producing, we run an animal experiment in Bioenergy. Bioenergy is one of the very few institutions in Indonesia that has the biosecurity level 3 labs. And we run that experiment with the aid of my former students, Indro Kahyono. He was here yesterday, but they left to Bandung today morning. They couldn't make it. Anyway, that's the isolator, and we inoculate the birds with the virus. That's my colleague, Professor Milton McAllister from the University of Adelaide, that he helped us for running the pathology work. The birds at the day 0 after challenge, day 1, and in day 2, almost the all birds died or very, very sick. And it was reflected to us that how the virus can kill like a big farm of, say, 800,000 or I don't know, 100,000 birds in one shed within 24 hours. That's the fact for that. We started to do necropsy. In the necropsy, the first thing that we found that the blackness of the breast meat, and within 24 hours, you can see the big differences in terms of the size of that. Like they lost the weight, almost 35 to 45% of the weight within 24 hours. Hemorrhages in the conjunctiva of the eyes, hemorrhages again in conjunctiva, hemorrhages in the proventriculus, hemorrhages in the brain, that clearly you will see that the brain hemorrhages, but in not that much of the cases. Very, very clear case of the blackness of the breast meat. And finally, we found that the bursa fabricius, that is responsible for the immune system, like the B cell immune system, is heavily affected by the virus. To see what's happened, we ran the histopathology. In histopathology, we'll see that in the A side, means the left-hand side, you will see the healthy, the bursa fabricius of the healthy birds compared to the left-hand side, which is almost nothing left, all dead cells and the liquid. To go deeper, we will see healthy cells at the normal spleen and the NDV challenge, almost all dead cells and a little bit liquid left, normal one and the dead one. We found very, very unusual behavior of the virus. The virus is a respiratory virus or the gastrointestinal virus. Why this virus invade the lymphocytes? It was a question that no one could answer it. We consult with lots of people in the world, and finally we ran an experiment. Here is almost all liquid. We ran the immunohistochemistry to confirm that losing of the cells, or we call it lymphocyte depletion, is caused by the virus. And in immunohistochemistry, we clearly showed all of those brown dots that you can see in here. They are viral antigens. I mean, the viruses kill the cells. Compared with the normal cells at the A and C, with the infected cells with B and D, you can clearly see lots of brown dots in the D representing the viral antigens. The other things is we use two different vaccines to protect them. The vaccine that exists in the market and the vaccine that we made. The vaccine that exists in the market is Lasota in the middle. If we challenge the birds with no vaccine, 100% of them shed the virus, of course, because they get sick. If we use the Lesotho vaccine, 80% of the birds shed the virus. Means they looks healthy, but they shed the virus. And those viruses cause the circulation of the virus in the different farms. And finally, they come up with an outbreak and they kill the birds. It is one of the reasons that we said, the genotype 7 of the NDV happens in the farm with the good biosecurity. The good biosecurity keep the birds healthy and happy, but the virus circulating there, and so finally it burst to kill them. The vaccine that we made, G7 vaccine, the level of the shedding is 25%. Means it can protect 70% of the birds from the infection. Anyway, the titer, if we use the heterologous titer, how do we tell the farmers that, this vaccine works or this vaccine doesn't work? We have to use the specific antigen related to circulating virus. If we use the antigen that is not related to the virus, the real titer is very low and it is not protective. If we use the antigen that related to the circulating virus, the level of the protection is much higher. If you put the cutoff point around 8, you will see that if we use the homologous antigen, it will protect the birds nicely. Homologous and heterologous in different groups. I'll jump over that. This paper is recently published by Putri, my former PhD student, who is the lecturer at the Nusa Cendana University. This afternoon, I'll fly to Kupang to visit Putri, and we will have a workshop for 2 days over there. This paper is published, and we clearly show that if we use the homologous antigen, we will get much better titer, much reliable results in here. The paper is published in Veterinary Journal. Anyway, to understand these disease production mechanisms, Dr. Noor talked about the microbiome. To understand the microbiome, we need to do the full genome sequencing. For full genome sequencing, in here, we use exactly the same technology, but rather than using the DNA as a source, we use RNA as a source. We call it transcriptomics. In transcriptomics, we will understand the disease production mechanisms for each disease. That's why the transcriptomics is the hot topic in the world. How to do that? At the first step, we isolate the RNA. In the second step, we make the cDNA library. For the third step, we sequence the cDNA library and we isolate them. If in PCRs, you can detect only one gene, in transcriptomics, you can detect up to 30,000 genes in one shot. Just imagine how much information you can get. And it is not a matter of the detection only. It tells you the level of the expression. Means they say this gene expressed 10 times, that gene expressed 1,000,000 times, that gene expressed 100 times, and that gene expressed 1,000 times. Means the level of the expression will tell us lots of stories. In here, when you compare at the left-hand side, the healthy birds with the diseased bird, you will see the genes that affected by the virus. We provided a list of 50 genes at the, say, left-hand side. Unfortunately, the pointer doesn't work in the screen. But anyway, you can see clearly the differences between the gene expression in these two groups, and we can say those genes are responsible. The next step is the network to see which gene has more effect, regardless of the expression, more effect, more, say, functional effects on the disease production. I can tell you, for example, one story. We have two different people in here. One of them is the commander, and the other person is a worker. One function by a commander can affect hundreds of the people. But one function by the worker affect very, say, limited. So in here, we not only detect the level of the expression, we detect the network. The network tells us which gene plays the central role for the disease production. A very a quick example, just turning on and off the lights in here, it is one function, but it has lots of effects, like lots of lights will be, or here might be dark, might be fell off, the TV will go on, and by one function. And in here, we detect, it is actually the entire project was almost for 7 years, and roughly $700,000 spent. 5 PhD projects, 1 honors project. The top right is Fong from Vietnam. Top left is Putri from Indonesia. Mohammed down, and Sami from Hong Kong. They are all helped together to create such a complicated that I never. Anyway, that is so beautiful. I'll put it in here. But in the middle, you will see some of the genes play a big role. What does it mean? If you want to protect a disease, you need to concentrate on those genes. For example, same as the microbiota. If you want to make people happier, if you want to make happier, you can offer them to eat some different food, if effect on the microbiota in the brain, and then people are happier, stronger, healthier, whatever. So anyway, Putri ran a very beautiful experiment. In here, we compare the genes that express in the response of the vaccine. Here is Lesotho vaccine, and the genes, we call it volcano plot. And the genes that you can see in here that highlighted in red or blue, they are the genes that responded to the vaccine. Lesotho has very limited genes of responding to the vaccine. I mean, the level of the protection. Just compare with the vaccine that we developed, G7 vaccines. In G7 vaccines, the numbers of the genes that affected by the virus is much higher. And all of those that we highlight them, even in red or blue, they are the genes that are responsible for the immunity. Means the G7 vaccine provide more stronger immunity. But the question is why the vaccine production companies do not make it? Because making of that is hard. It is not economic. So the reason we found some other reasons that the virus that can kill the host. The most important one is apoptosis, and apoptosis is programmed cell death. Means the virus initiate that and the cells will die. The other things is DNA degradation. We ran, say, another experiment, and we found that the DNA in the hatch, the DNA is degraded in the response to the virus. Means the virus directly destroyed the DNA of the host cells. The paper is published recently in Nature Scientific Reports. For your information, because probably you are interested to publish a good, say, good value the papers. I'm the editor for the Nature as well. You could mention it in my CV. I'm the editor for Nature Scientific Reports. Actually, we submitted few papers, and the paper is rejected. Even I'm the editor for the journal. I don't have influence to publish even my papers. The papers needs to go to the proper channel. Anyway, the paper is in Nature is published by Muhammad. And Muhammad discovered that another terminology. We have a term of necrosis and apoptosis, and we created another terminology as necroptosis. And the necroptosis is new pathway that can kill the host cells. Anyway, I'll go to the other. Another one is, I don't know how many minutes do I have. I think 2 minutes left. I think it is the very last slide. In here, in all of the papers that publish on transcriptomics and the network analysis, people concentrate on the host genome. And the reason for that, we have million genes in our cells, but the virus has only 7 or 6 genes. Means the genes of the viruses will be diluted or even disappear. No one can see them within like 7 genes compared to the 100 million genes. So this time I read the paper published in Nature. Probably this paper is the best ever paper that published for the COVID. I would suggest you, if you want to understand the disease production mechanisms by COVID virus, read that paper. I can put the link. But when I read that paper, I talked to Fuang and I said, "Hey, Fuang, you need to analyze the virus genome." And she said, "No, Farhid, only 6 genes. No one can publish it for us. No, no, no." And I said, "Yeah, anyway, you publish. Anyway, it takes 3 months to prepare all of those materials and write the paper." And surprisingly, the Nature Scientific Reports publish it for us. It is the first paper in this field that we can say how the viral genes, I just highlighted them, how the viral gene interact with the host genes. In all of the others, the host genes interact with each other. In here, the viral gene interact with each other. We can easily say how they're interacting and how they produce diseases. Anyway, as a conclusion, we can say that Newcastle disease genotype 7 is highly pathogenic virus causing the deadly infections in the birds. We have to use the proper, we have to use that I had a seminar in UB and I asked them that, is there any G7 antigen exists in Indonesia? They said, unfortunately, no. Even G7 is originated from Indonesia, means your country is own the strain, but you still do not make it. It is a little bit pity that making that antigen is hard, is not simple, but as a pioneer in the world, you can make it. So I want to say thanks to the people who work for me. From the left-hand side, Risa Indriani from the Balitvet, Ningshi, my former PhD student in Balitvet, in the Darmentary, who is the big boss under the president of Indonesia. She is the director for BRIN. She was my former student. Anyway, I have a good relation with Indy, Simpson Tarigan, and the Esther Yip from Hong Kong. They work, and in the down, I'll put the people in my group. And finally, when I proposed it last year in the Nusa Cendana University, a group of the students came and said, "Yes, we are interested in this project," and probably one of them will be join my group in next few years to continue the same project. Thank you very much for your attention.